Students, in this lecture, I'm going to talk about adaptation to stationary phase in a general manner. Like these uh, steps are very much similar to different kinds of stress. It can be nutritional stress, it can be heat stress, it can be um, shock stress, osmolarity, salinity, or oxidative stress. The way the cell responds, these are the common things which you can write for all these stress. But in case of cold stress, there are specific alterations which I mentioned in the last lecture. These are the common adaptations of cells to the stationary phase. So, bacterial growth can be divided into five phases or five areas of growth. The first is the lag phase. La- Lag phase is the phase where the adaptation of the cells to new environment happens, where you have a lot of nutrients and the cells will start acclimatizing themselves with the new environment and start growing. And then you have the exponential phase where you have the rapid growth, and then comes the stationary phase. Stationary phase is where there is a lot of changes happening there will be secondary metabolites produce the cells, there will be nutritional depletion that will lead to slowing of the growth as compared to the exponential phase. And then you have the death phase where you have no nutrients available from the nutrient media, and the cells start dying. By the end of the death phase, that is, most of the cells would have died and thereby contributing to nutrients into the nutrient media. You have the long term stationary phase where you have the cells which are not dead, which are survive, they become resistant cells or dormant cells which can come out of this dormancy, and then they can start reproducing and growing faster once the conditions are provided. So this long term stationary phase has many sub phases if you provide the required conditions. So stationary phase is characterized by many of the um, important things, important points. Like in stationary phase, I told they are all dormant cells, that means they are not dividing, but there is growth. That growth is constant and in a slow pace. That means proteins are being synthesized at the constant rate, very slow rate, and then they are being degraded also in a very slow rate. So this alteration, that for protein synthesis you should have the mRNA, that means there is alteration in the metabolism of the whole cell. That metabolic change or acclimatization to this new state, that is long term stationary phase, is done by changing the transcription machinery. Now when you have transcription happening, you have the RNA polymerase recognizing the promoters, and then you have there is production of mRNA and proteins, and use of those protein in a particular requirement. So the primary thing the cell does is the change in the transcription machinery, thereby implementing the morphological changes. Instead of going to direct morphological changes, I am starting with changes in the transcription machinery. That is, stationary phases is characterized by accumulation of specific sigma factors. Now normally, when there is transcription happening, this RNA polymerase is a multi subunit protein, and this multi subunit protein is common to all the promoters which are recognized by RNA polymerase. But the only change which changes the expression of these promoters in bacteria is the sigma factor. Sigma 70 is the housekeeping a sigma gene and a sigma factor. So that is RPOD, that it is also called as sigma 70. um, it is like normal housekeeping genes are produced when this sigma factor is expressed, but there are specific sigma factor like sigma 32 which is expressed in heat shock, or you have sigma s which is produced under some stress conditions. So there are basically four group of sigma factor. Group 1 belongs the housekeeping gene, group 2 belong to specific functions like sigma 54, group 3 also belong to some specific functions, group 4 has this stress induced sigma factors. So these sigma factors, for example, the RPOS is one of the sigma factor which is gets accumulated on the onset of the stationary phase. This sigma factor directly or indirectly influence lot of genes in E. coli. Around 10% of the genes are influenced by this RPOS sigma factor. So the entire cellular physiology changes when you have this accumulation of the sigma factor RPOS. There are a lot of this RPOS dependent genes. The genes that belong to this RPOS regulon are involved in morphological changes of the cell, resistant to various stress condition. Example: oxidative stress, heat stress, osmotic stress, near UV radiation, or pH changes. There is also metabolic processes which are induced by this RPOS dependent genes that helps in adaptation to stationary phase, virulence, and the GASP phenotype. Okay, we'll see later what is GASP phenotype. So, bacteria in stationary phase undergo morphological changes, that is, cells become smaller as a result of two processes, that is, reductive division and dwarfing. Reductive division is like increase in the reductive division increases the surface area to volume ratio, producing spherical cells. Also, the expression of certain very specific transcriptional regulator, for example BOLA, which is like uh, which is very important because it controls the RPOS synthesis. Dwarfing is a form of self digestion and is the result of degradation of endogenous cell material, especially from the cytoplasm and outer membranes. So, in stationary phase, there is production of cell envelopes for effective protection of the cell against different kind of challenges. The transformation is to enhance the barrier induced by these external changes in all structures of cell envelope like outer membrane, periplasm, peptidoglycan, and inner membrane. So the concentration of lipopolysaccharide will increase in the exterior surface of the outer membrane. There is also reduction in the amount of protein in the outer membrane, together with increased cross-linking of outer membrane lipoproteins and peptidoglycan layer. The periplasm accumulates membrane-derived oligosaccharides such as trehalose, which has an important function as osmoprotectant. The peptidoglycan layer, which is also serves as a stress-bearing component of the bacterial cell, increases in thickness. So there is increase in synthesis of D amino acids during stationary phase, and because they modify the peptidoglycan, they are important for peptidoglycan layer. They modify the peptidoglycan layer by their incorporation into peptidoglycan polymer, and repression of peptidoglycan synthesis also happens. The inner membrane also undergoes multiple changes like decrease in monounsaturated fatty acid. There is also increase in polyunsaturated fatty acid. Unsaturated fatty acids are converted into cyclopropyl derivatives, and the ratio between phosphatidylglycerol and phosphatidylethanolamine increases as cells approach the stationary phase. So, as a consequence of this, the inner membrane presents a highly ordered structure with reduced fluidity. There is alteration in cell envelope occurring together with changes in cytoplasm. The nucleoid become condensed to protect the DNA. That is, there are a lot of DNA binding proteins which are synthesized, especially these proteins were observed in starved cells. That is, one of the important protein is DPS, because it is a non-specific DNA binding protein that preferentially acts during starvation. This protein is present in various bacterial species. Around 130 bacterial species express this protein, and this expression is modulated by sigma 70 dependent under oxidative stress by factors such as OXYR and RPOS, which are also seen under starvation stress. So when this DPS protein becomes more abundant, they form ring-like dodecamer structure, uh, like they bind to DNA when magnesium is present. So when they bind to this DNA, they form a crystal-like structure. It is also called as biocrystals. This results in compact nucleoid. So DPS also shows significant structural homology with other proteins like ferritins, which have double functions of sequestering iron ion and reducing the formation of oxidative stress. There is also dimerization of ribosomes that could be responsible for the decrease in protein translation. That is, dimerization of this ribosome um, requires a ribosome modulation factors that cover the peptidyl transferase domain and the entrance of peptide exit tunnel, blocking the ribosomal translation activity. So there are different proteins which get associated with ribosome. Like one of the protein is RMF, which is induced by phosphate guanine. This pentaphosphate guanine is produced more and more under stationary phase. There are also other proteins like YHBH, which promotes the and stabilizes ribosome dimer formation. There is one more protein called YFIA, which prevents 70S ribosome subunit dimer formation as stationary phase is induced. This ribosome associated protein and a hibernation promoting factor. It functions as a ribosome associated protein and hibernation promoting factor. So, dimerized ribosome could represent a way to store ribosome during periods of translation inactivity. There is reversion when the conditions become optimal for growth within few minutes. During entry into stationary phase, there is expression of one protein called as FADR. This full is expressed by FAD operon. That is, there is this the expression of the FAD operon is controlled by the repressor, that is FADR. So when there is increased production of this FADR, these FAD operon is regulated. That is, FAD operon is required for growth using long chain fatty acids as carbon source. So there is fatty acid generated from degradation of a membrane lipid during the dwarfing process that could be scavenged by the activity of acyl CoA synthase to generate acyl CoA, which could be metabolized by beta oxidation enzyme, and thereby generating carbon and energy. There is also increased synthesis of glycolysis enzymes like pyruvate formate lyase, phosphotransacetylase, and acetate kinase. Together with a lower production of TCA enzymes, which mediate a three component regulator system like ARCB, ARCA, and RSSB. There is also repression of aerobic metabolism upon entry into stationary phase, which may prevent the uncontrolled use of endogenous reserves during conditions like autophagy, and it can also be a defense mechanism in starvation against the damaging cells to which that is against damaging effects of reactive oxygen species produced by respiratory chain. Under stationary phase, there is also a production of QS molecules. These QS is a cooperative behavior that coordinates gene expression in a cell density dependent manner as bacteria leak signaling molecule which accumulate depending on cell number. So when this concentration of QS becomes more and more, the cell detect it and trigger a specific response that may be important for the formation of biofilm and virulence. Again, the sigma factor RPOS controls this QS gene, which is like onset only in the stationary phase. During stationary phase, where wide collection of secondary metabolites, antibiotics, and toxins are synthesized, there is molecules like micro sins, that those are very small bacteriocins with a strong antibacterial activity against closely related bacteria, are synthesized that are helping to fight between intestinal microbiota and foreign enterobacteria normally. So the production of this sigma factor RPOS also controls various expression of virulence factor in a normal in human pathogens. So under stationary phase, there are a lot of changes happening. That is morphological changes like the cells become smaller and spherical, the nucleoid get condensed. Metabolically, there is a stringent response. Only what is required is synthesized in the stationary phase. There is repression of aerobic metabolism, thereby reducing the oxidative stress. There is increase in the fermentative enzyme expression, production of ribosome modulating factor. There is drop in protein synthesis while increase in peptidase and protease synthesis happen. The change in sigma factor affinity also happens. There is translational regulation by forming 100S ribosome dimers, which decrease the protein synthesis, and other factors like which help in increasing resistance to physical and chemical stresses like synthesis of quorum sensing molecules or production of secondary metabolites. There is programmed cell death of certain cells. You have the production of GASP phenotype. Now what is this GASP phenotype? These GASP is a condition where the ability of the aged cells to take over the young cells happen. So a specific changes happens in this phenotype, that is like the stable mutations happens under this phenotype, which helps the cells to survive the starvation. During the stationary phase, there is continuous cycling of growth and death in starved cultures occur due to rise and propagation of mutants with greater fitness than the parental strain. The kinds of mutations what happens in this GASP phenotype is one changes we normally when the cells are entering the stationary phase, there is production of sigma factor RPOS. Now there is a mutation happens in this RPOS factor, sigma factor, that is the last C terminal 4 amino acids are replaced by 39 new amino acids, so thereby stabilizing more this RPOS. And there is the physiological role of this RPOS leads to adaptation to the stationary phase. So there is one more mutation which is also mapped in this GASP phenotype, that is LRP gene, which encodes for leucine responsive global regulator. There is mutation observed. So this mutation which is seen is responsible to cause a in-frame 3 base pair deletion, originating a protein lacking a conserved glycine residue, that is G39, within the helix turn helix domain. So there is loss of function mutation behaves as a dominant negative, meaning that the stable non-functional LRP can still form dimers and with a wild type monomer blocking its activity. So this dominant negative mutation can change the cell physiology, uh, which seems very important, especially under starvation mutants. Additionally, especially in E. coli cells, there is RPOS down mutants when they are like uh, observed. They shows mutation in BGL operon also. This BGL operon is involved in uptake and use of aryl beta glucosides, glucoside, salicin, and arbutin. So when there is activation of this BGL operon, uh, and then in the parental strain there is stability. There is also activation of point mutation in CRP CAMP binding site in regulation region of the operon, which helps the GASP phenotype to prevail. There are also other phenotypes like VBNC, that is viable but non-culturable state, which are produced. The like certain bacterial cell species under prolonged starvation lose their cultural ability, failing to grow under standard laboratory condition, but still they remain viable. So this condition is known as viable but non-culturable state. So under this state, there is very low metabolic activity, and the same changes of a stationary phase like dormant stages is observed in this VPNC condition. There are also other phenotypes like stationary phase constant dependent inhibition phenotype, which is produced, that is after several culture experiment, especially in E. coli K12 strain. These emerging strain SCDI emerging strain were observed, and they were known to kill or inhibit the parent strain. So this inhibition ability was due to several single base non synonymous substitution in GLGC gene. This encodes for a ADP glucose pyrophosphorylase, which is a regulatory protein involved in glycogen synthesis. So the more of glycogen synthesis is produced, which is also a stimulant for the survival of the cells under stationary phase. So in stationary phase, there are a lot of changes happening, and production of new phenotypes are produced under the stationary phase.